Hi, my name is Ann Wheeler, and I'm providing a recording here of a presentation that was originally given by my colleague Sarah Andrews at the 5P- Society Conference in July of 2025. I'm going to be going through these slides and talking about the importance of disease concept models and natural history study participation for ensuring that therapeutic development in rare disease prioritizes what matters most to patients and caregivers. First, I'd like to provide a bit of background on who we are and why we're providing this information for this community right now. Sarah and I are part of a larger multidisciplinary team of clinicians and researchers who focus on supporting rare disease communities, advancing earlier diagnostics, and promoting effective therapeutics. I am a psychologist by training and focus on clinical outcome assessment development and validation, understanding developmental and behavioral trajectories, and supporting the development of resources for families. I'm also a clinician and support both assessment and consultation around behavior and development for parents and individuals with rare disease at the Carolina Institute for Developmental Disabilities at UNC Chapel Hill. Sarah is a close colleague of mine who I've worked together on multiple projects. She is a qualitative researcher who focuses on ensuring lived perspectives of individuals and families who are impacted by rare disease are integrated into all research efforts. Sarah and I work at RTI International. It's a nonprofit research institute whose mission is to improve the human condition by turning knowledge into practice. Our team at RTI was connected to the Chat Research Foundation by word of mouth from other rare disease communities we've worked with. The CDCRF was founded by Megan and JC Leston after their son Liam was diagnosed with 5P-. Their mission has been focused on developing therapeutics, meaning medications and things like gene therapy, to improve the quality of life and the level of independence of children and adults living with 5P-. The CDCRF has a strategic roadmap focusing on 3 pillars of research efforts to promote therapeutic development for 5P-. The first is to distinguish which missing or non-functional genes play a critical role for individuals living with 5P-. The second pillar is focusing on replacing the missing or non-functional genes in the neurons of individuals living with 5P-. And in order to get to the place of being able to do clinical trials that will help push forward treatments for fixing those targeted genes, we also need to make sure that we are focusing on accelerating and preparing for clinical trials. This third pillar represents the actions that are needed to allow all of the work in pillars 1 and 2 to reach individuals who are living with 5P- syndrome, and to ensure that transformative therapeutics are advanced in a healthy and effective way. Our team at RTI focuses mostly on supporting this third pillar. Part of the CDCRF's translational research plan involves the development of a natural history study with appropriate outcome measures that can be used in preparation for clinical trials. Over the next few slides, I'm going to walk you through the importance of this effort and the steps we need to take to make sure this study gets off the ground in a way that is most effective for the community. To begin to describe this process, I want to zoom out for a minute and talk a little bit about the full picture that's involved in the development of a therapeutic drug for 5P- and other rare neurogenetic conditions. Therapeutic drug development requires a multi-pronged approach that includes basic science research to identify the genetic mechanisms associated with a condition. Once identified, scientists will replicate the genetic changes in both neuronal cells derived from patient cultures, as well as through development of animal models where they can simulate behavioral symptoms that are often seen in patient humans with the condition. This allows drug companies then to be able to target the symptoms that are necessary and needed for therapeutic development. It becomes very critical then that we have good understanding of what the condition looks like and what the priorities are for treatments, so that once we get to this place, we know exactly where we want to target. This infographic, published by RareX, another organization that supports rare disease research, describes 4 categories of need for a rare disease community in preparing for the possibility of a disease-modifying therapeutic. The first is to understand the disease biology. We need to not only know what the causal genes are, but also understand the biological function of that gene and what happens downstream when there are variants within that gene. We also need to understand if there is therapeutic potential. Does the gene change lend itself to the possibility of a target for drug development? Can that be initiated in a safe way? And can it be precise in its targeted treatment? We also then need to have the community be clinical trial ready. This means we need to understand what biomarkers exist that can effectively tell us whether or not the drug has had an impact. We need to know what the clinical trial endpoints are as far as what would be meaningful to patients and caregivers. And we need to have enough people out there who understand the condition that can be part of a clinical trial and make sure that we're doing the due diligence to measure change over time. And of course, we need the population to be engaged and interested in participating in clinical trials and moving the condition closer to a therapeutic. The first 2 steps in that infographic are actively happening now. 5p- is a complex condition with multiple genes involved. Still, there are a number of potential directions for therapeutic development that are being considered by basic science researchers around the world. As a result, it's very important for the community to start thinking and planning for the possibility of therapeutics that could potentially have a positive impact on the quality of life for individuals with 5p-. So we want to make sure that we're narrowing in on these 2 other boxes at the same time that the more basic science work is being done, so that we can hit the ground running when there is a potential treatment ready for trials. According to the FDA, in order to be ready for clinical trials, we need to have a thorough understanding of the condition, which will include understanding the natural history of the condition, what kinds of subpopulations might exist within the population of individuals who have the diagnosis, what current treatment practices are, and most importantly, what patients and caregivers expect and hope from a potential treatment. Once we understand the condition, then we can start to develop models for what would be of most clinical benefit and start to develop tools that can measure any potential changes in those domains of need over time and in response to treatment. In order to do this, we want to start with a disease concept model. That disease concept model will help inform the outcome measures we choose and the ones that we put into a natural history study. The natural history study then, using those outcome measures, will be able to track what the condition looks like without any active therapeutic. And this will establish the baseline data that's absolutely critical for us to understand if a potential treatment in the future is actually making a difference in the quality of life of individuals with the syndrome. So what is a disease concept model? A disease concept model is a framework more than anything else, and it represents how we think about many aspects of a disease. Most importantly, we want to be able to describe in as much detail as possible what the symptoms are for the condition, how it impacts the affected individual, and how it impacts those who are supporting them, including their families. One of the most important elements of developing a DCM is to be able to interview families and patients about their experiences and how their symptoms affect their daily life. As I noted, there are 3 major components to a disease concept model. The first is to understand the symptoms. As much as possible, we want to understand the symptoms from the patient's point of view. We want to understand how the symptoms affect their life, and we want to understand also how their symptoms affect the family's life. From there, we can extrapolate how those impacts affect the individual's activities of daily living, so how they move about their day, as well as their school, their work, any care that they receive, and their social networks. As far as impact on family, we want to understand what the burden of the disease is from a financial and an emotional standpoint, as well as how it might be impacting the community the individual is living within. And from these, we can then start to identify those endpoints that will be the critical assessment of whether or not any potential treatment will work and is considered effective. The disease concept model will not only benefit future clinical trial designs or the ability to understand whether a treatment works. Having a disease concept model also improves communication and collaboration among clinicians who might be working to provide the most patient-centered and effective care possible. It's also really important to understand the lived experiences so that all elements of treatment and care going forward are centered around the experiences of the patient and their family members. There are several steps that we need to follow to develop a disease concept model that will be useful in supporting clinical trial readiness. The first is typically a literature review. In this literature review, we would look at what has already been described in the history of 5P-, including things like what are the primary symptoms that parents and individuals have experienced? What are the most burdensome things that individuals and families have experienced? What current treatments exist? And what concepts have previously been captured to understand the condition? Then from there, we would take that literature and develop an initial concept model that would be then shown to families and individuals with lived experience, where we can gather their feedback on what of these domains that have been reported in the literature should be priorities in future clinical trial therapeutic development. Fortunately, this process has already been started. This figure represents an initial disease concept model that was published by a team of investigators in Spain who mapped out the most common symptoms seen in 5P- using clinical records and questionnaires from 70 patients. While this is a critical article that sets the stage for understanding the condition, they did not take into account which of these symptoms are most impactful for the individual or family and which symptoms should be priorities for future treatment development. More recently, Ayesha Gurujala, an undergraduate student of Terry Jo Bichelle, who is a neuroscientist and the founder of a nonprofit organization called Combined Brain, did a literature review and mapped 5P- symptoms to an initial disease concept model. In the first part of this model, they mapped out the defining concepts, essentially all of the symptoms that are reported in the literature within specific domains of functioning. Specifically, all symptoms are mapped out to be part of communication, neurological, cognition, emotion, behavior, motor, skin, or visual set of symptoms and domains. In addition to identifying the core concepts and symptoms that are important for the individual, they also mapped out an initial set of individual impacts as well as caregiver impacts. For the individual impacts, they looked at things like how the symptoms might affect the individual's functioning within the community and school, within socialization with peers and their family life, and within their daily living and self-care. They also looked at impacts that were likely to have an effect on the caregiver, including things like the need to be at home to care for the individual, sustained financial concerns, and being able to connect to other families. And they also considered modifying factors that may have an influence on the extent to which the individual symptoms may impact the caregiver. So this work provides a really important foundation in order to better understand what the true lived experience is for patients with 5P- and their families. This is an initial disease concept model that is based solely on the literature and requires some feedback from families before we can move forward with it as the official disease concept model. So the next step, which we hope to be initiating in the near future, is to take what Aisha and Terry Jo did in their initial disease concept model based on the literature and understand from parents, patients, caregivers, and clinicians what is missing from this initial model and how we should think about priorities within all of these different symptoms that may have an impact with regards to what we need to focus on for treatment and what they feel are the most impactful symptoms for their quality of life. This is an example of how we might take the initial disease concept model and map it against the things that we hear from families in qualitative interviews. This is an example from STX-BP1, another rare disease condition, and it describes what we can do ultimately with the qualitative interview data. So here we see at the top the symptoms. So again, that's that first slide that I showed you that had all the different boxes of potential symptoms that affect individuals with 5P minus. So we would be listening to interviews to hear how much of each of those symptoms are families talking about within those interviews when we ask them about priorities. We'll also be listening for the impact that those symptoms have on the individual, as well as the impact that those symptoms might have on the caregiver. And you can see here we get some quotes about how those things are actually having an impact on the individual's daily life. We can also use things like heat maps to allow us to understand the extent to which those symptoms are impactful and the extent to which they may change over time. So here on the right side, we have the symptoms that are mapped out on the left according to the age of the child. So you can see things like an infant in toddlerhood, seizures and issues with tone might be most important and impactful, but as an individual gets older, things like behavior, cognition, and expressive communication become more impactful. And just to point out, this particular disease concept model is based on over 25 hours of interviews with a number of individuals, and there were, in this set of interviews, over 3,000 references to 38 distinct concepts. So you can see how this interview method can provide really rich and useful data for the future. So once we have these symptoms, we can use this interview data to map the most important priority symptoms and think about how those symptoms might be different across the lifespan. This is just a blow up of the slide I showed before with some of these additional quotes. Here's an example of a finalized disease concept model for another condition called Angelman syndrome. And I want to talk a little bit now about how we take this disease concept model and take it to the next step to be able to map outcome measures. So this model was developed in the same way as the model that I was just showing you, and in the same way we are proposing to develop one for 5P minus with parent interviews and literature review setting the stage. So you can see here the defining concepts as we had described previously, and then we have the individual impacts and the caregiver impacts. So what Angelman syndrome has done is they've taken all of these defining impacts, and the two patient advocacy organizations in Angelman syndrome called the Angelman Syndrome Foundation and the Foundation for Angelman Syndrome Therapeutics came together, and they developed a third independent organization affiliation called the ABOM. This is standing for the Angelman Biomarker and Outcome Measure Alliance. And so they co-funded this effort and brought together a team of experts to think through each of the defining concepts and what outcome measures exist and what outcome measures need to be developed in order to appropriately and accurately and sensitively measure these priority domains. It's important for us to take a step back and talk a little bit about why outcome measures are important and why this effort to really think about and consider the most appropriate measures is absolutely critical. So it may seem really simple. We just ask patients and caregivers what their experiences are. But we have to be able to indicate and show that each outcome measure is objective. And we have to think about things like, does the measure reflect the lived experience of patients? Can all individuals with 5P minus complete some of the tasks required by a specific measure? Do the measures identified have solid evidence of being valid, reliable, and sensitive to change? And these are all things that we need to be thinking about across the different types of outcome measures that exist. So this slide illustrates the different type of measures that are typically used in clinical trials. Performance-based measures, outcome measures, are basically what it sounds like. These are direct assessments of a patient, and it's assessing a specific set of skills that the drug or treatment is considered likely to have an impact on. So examples of performance-based outcome measures might be a walking test. It could be a direct assessment of cognition or development, like the Bayley scales of early learning, or a WISC cognitive test. And the challenges with performance-based outcome measures in a population of children with intellectual or developmental disabilities is that these measures need to be ones that, again, any individual with a condition can perform at least some elements of in order to be considered a valid measure for that population. Another set of measures are patient-reported outcome measures, and these are tools that ask the patient directly to report on their symptoms. And as you can imagine, patient-reported outcome measures can be very difficult for individuals who have language or cognitive impairments. And so therefore, we often turn to observer-reported outcome measures to replace patient-reported outcome measures in clinical trials in which an individual cannot self-report. So observer-reported outcome measures are typically completed by caregivers, such as parents or teachers. And while often considered more valid than patient-reported outcome measures in these types of trials, they also have inherent challenges of being strongly skewed by placebo and may have rater bias. So for example, a caregiver who is highly motivated to see a change in their child may see a change in their child even if there isn't objective change. Similarly, we can have challenges with recall. If something has happened recently, individuals may be more likely to remember that recent event than to consider all the different types of events that the questionnaire is actually asking about. And then another important component of all of these, last type of outcome measure is clinician-reported outcome measures. And these would be ones that a doctor or another professional would complete about an individual after spending time with them or observing them in some setting. So all of these measures, types of outcome measures, are critical to be able to understand the full extent of a treatment. And a really important element of all of this is that in order to be used in a clinical trial, the FDA has to approve the use of that measure as an outcome measure in that particular trial. And in order for the FDA to approve the measure, we have to be able to show solid evidence that the measure is valid. In other words, the measure needs to accurately measure what it says it measures or what we are asking it to measure. Measures need to be reliable, which means that there's consistency in the measure regardless of who administers it or when it's administered. The measure has to have strong psychometrics, and this means that all of the items need to be measuring similar things, and that also that there's a range of difficulty within those items so that we can identify differences between patients. Finally, this is an especially important point for clinical trials. The measure must be sensitive to change. And so we want to make sure that as the measure is being used over time, we can be confident that if there's change in the measure, that it is due to an intervention and that change is clinically and meaningful to that individual. So in order to make sure that all of the outcome measures that might be used in a clinical trial are valid, reliable, psychometrically sound, and sensitive, we need to collect a lot of data on those measures. And so that's one of many reasons why a natural history study is absolutely critical for 5P minus. So other reasons we need to have a natural history study is that we have to be able to identify the trajectory of the condition without treatment. So we need to know if nobody does anything or if we do things that are basically the current standard of care, what does the condition look like? How does it change over time with age, etc.? And then that can help with determining in the future what a prognosis might be and give information for individuals, caregivers about what to expect. And then within a clinical trial, having a natural history study can help us determine, again, if a treatment is really effective. It can provide important information about how well a measure works and how much time we need to have in between assessment times, how much time we think it will be important to have between when a therapeutic is delivered and when an assessment is conducted, and how long we think the trial should go. In some cases, and this is a little bit tricky to get approval to do, but in some cases it's possible that if a well-designed natural history study can provide a control group for a clinical trial, which will reduce the need of having a placebo arm. So natural history studies are really important, and they're critically important for rare disease in a space in which a potential clinical trial for a therapeutic is in the pipeline. So this is just another little infographic that I think is helpful to understand natural history studies and another resource for families and individuals who might want to know more. So in addition to being able to collect this data that can help us with supporting drug development, the more we know about a condition and the way it changes over time, the better we can be as far as developing and improving the standard of care practices that are used in clinical treatment. So if we understand, for example, that a specific medication or a specific behavioral therapy has benefited children in the past, that can be something that gets integrated into recommendations for younger children who may be coming up behind them. So what does all this mean for you? So as I have laid out, as we've tried to summarize here, we have a pretty good start to the first few steps. We have an initial literature review and an initial concept model that was put together thanks to Combined Brain. And now we need to take it the next step. So we need to understand how clinicians and caregivers prioritize symptoms based on that initial concept model. And then we need to finalize the concept model and identify what the important priorities are for a natural history study and identify initial measures to be used going forward. It's currently a very optimistic time for individuals with genetic conditions as more and more science is developing disease-modifying treatments that have the potential to improve the quality of life for individuals and their caregivers. So the Kruduchat Foundation is looking to fund this effort and move this work forward into these next steps. We will need you, the individuals with real-lived experience, to step in and be citizen scientists and provide us with the information that we need to be able to move forward with this. So we are encouraging you to get involved when it comes time. We will be looking for families to participate in interviews and eventually to participate in the natural history study. And just a really strong reminder that without you as the individuals with the real lived experience of this condition, we can't move this forward. So if you'd like to learn more about what we're doing here and hopefully eventually contribute to this work in the future, we would love to hear from you. If you have specific questions about disease concept models, about this presentation, or you want to learn more about, in general, about disease concept models and natural history studies, we are more than happy to answer your questions and have a conversation with you. I've left my email here, which is acwheeler@rti.org, and my colleague Sarah's email is sandrews@rti.org. Please feel free to reach out to either of us. If you know you want to participate in an interview about your family's experience, please do reach out to Megan Leston. She's collecting names for people who we might reach out to if and when we're able to get this project funded and off the ground. Thank you so much for your attention to this presentation, and we look forward to working with this community in the future.